Good day, grade 12 learners, and welcome to this video. We will be focusing on the process of meiosis. You would have remembered in the previous video we did a brief introduction to meiosis, and we will now looking at describing the entire process of meiosis. In the previous video, we revised the structure of a cell with emphasis on parts of the nucleus, the centrosome, and the cytoplasm. We looked at the structure of chromosomes. We also differentiated between haploid and diploid cells in terms of chromosome number. We spoke about sex cells, and we also spoke about sex chromosomes known as gonosomes, as well as our autosomes, which are our body cells. We also looked at the division of the process of mitosis, which was the cell division that was dividing for the simple purpose of growth and repair of damaged cells, which was what you would have covered in your grade 10 syllabus. In this video, we will be focusing on the entire process of meiosis. So firstly, we will define what meiosis is. We'll speak about the site of meiosis, namely in plants and in animals. We'll speak about meiosis being a continuous process, but the events are divided into phases for convenience. We'll describe all the events that occur in interphase. We will then look at the events following the phases of meiosis I using diagrams. We will describe prophase I, metaphase I, anaphase I, as well as telophase I. Firstly, let's try to define the entire process of meiosis. Now, in grade 10, the normal type of cell division that you learned about was mitosis. And you'd have learned that mitosis, the purpose of that was simply for the growth and division of normal body cells, and you were going to divide that cells for growth and repairs of damaged cells and damaged tissue. But in meiosis, it's going to be completely different. So meiosis is a special form of cell division to form sex cells or gametes, and it occurs in reproductive tissue of plants as well as animals. Meiosis is referred to the type of cell division where you are going to be forming sex cells or gametes. And when we do this, we are simply going to be halving the chromosomal number. So originally, you had 46 chromosomes in your normal somatic cell. But after meiosis, you are reducing your chromosomal number to half. So in human beings, it's going to move from 46 chromosomes down to 23 chromosomes. If we talk about the site of meiosis in plants and in animals, now you would have learned last year in grade 11, when you spoke about plant diversity, you would have focused on the reproduction of each type of plant diversity, such as bryophyte, pteridophyte, gymnosperms, and angiosperms. And you would also spoke about the reproductive structures that you have in each plant. So if we have a look at plants, so very similar to how you learned in grade 11, your male cells are going to be referred to as the pollen grains. And inside the pollen grains, that's normally where you have your male gametes or your male sex cells. It's formed in the anther because the anther is what produces the pollen. Supported by the filament, the anther and the filament together form the stamen. So just a reminder, the male reproductive structure found inside the plant is referred to as the stamen. But the anther that is found at the stamen produces the pollen. And in the pollen, you're going to have your male gametes. So this is what the stamen looks like. Very similar to what you dealt with in grade 11. If we look at the female part of the flower, the female cells are referred to as the ovules. It's formed in the ovary and at the base of the style. So therefore, the female reproductive structure is going to be it's going to comprise of the stigma, the style, and the ovary. So it's going to be referred to as the pistil or the carpel. Just an example of what your male pollen cells look like, and another example of what your female ovule cells look like, because the ovules are going to be formed inside the ovary of the female. So these are your pollen grains on the left, and these are your ovules found inside the ovary of your female reproductive cell found inside your plant. If we now just have to look at the human life cycle, because remember when we're describing meiosis, we are focusing on meiosis firstly in human beings. Before we're able to understand anything else, we first need to understand meiosis in human beings. So let's look at the reproductive life cycle that occurs in any human being. So on the left-hand side over here at the bottom, by the red section, you have a male and a female. And you know that males and females should have their own reproductive organs, but they both undergo a process of meiosis. And when they undergo meiosis, it's a type of cell division where they are halving the original chromosomal number that was found in a somatic cell from 46 chromosomes to 23 chromosomes. If we look at the male, when the male undergoes meiosis, he is going to form the sperm cell, because the sperm cell is known as the sex cell that comes from a male. When the female undergoes meiosis, the type of gamete or sex cell that she forms is going to be the egg cell. And you see that the chromosomal number now becomes 23, where originally at the start, the somatic cell comprised of 46 chromosomes. When that sperm cell and that egg cell are going to fuse in the process of fertilization, eventually they're going to form a diploid zygote. And that diploid zygote now first needs to undergo mitosis. It needs to grow into an adult male or female, and then you are going to start undergoing meiosis, and the cycle is going to continue. So this is just a brief look as to where we are actually heading to in this topic. After we have our gametes or sex cells, where are we actually going to? So it's not something you really need to grasp yet, but in the next topic, reproduction, we'll be coming back to this, and then this will make a lot more sense to you. We will now move on to explaining a very, very important concept, a terminology question that you are required to understand. Now remember, we said that meiosis is going to be a type of cell division that's going to halve your chromosomal number, and the purpose is simply to make your gametes or your sex cells. Now meiosis generally occurs through a process, and that process is referred to as gametogenesis. You need to know this word, it's very important. The process of gamete formation is referred to as gametogenesis. And you have two types of gametogenesis. You either have gametogenesis taking place in males, or you have gametogenesis taking place inside females. When gametogenesis takes place inside males, it's going to form male gametes, referred to as sperm cells. This process will occur inside the testes. The testes is going to be seen as the male reproductive organ that's going to be found in human males. In females, when females undergo gametogenesis, they are going to be forming female gametes, which are referred to as the egg cells or ovum. And this is generally going to occur in the ovaries, because the ovary is known as the female reproductive organ. So three things you need to remember here. Firstly, the name of given to meiosis, the process of gamete formation, is referred to as gametogenesis. It can occur in males, where it's going to take place inside the testes in males. That's the male reproductive organ. The name given to the gamete that it forms is going to be sperm cells. If gametogenesis occurs in females, it takes place inside the ovary. The ovary is the name of the female reproductive organ, and the name of the gamete or the sex cell is referred to as egg cells or ovum. We are now slowly moving over to describing the process of meiosis. But before we do that, let's just take a brief recap as to exactly what happens in the structure of a chromosome. Now, you'd have remembered in the previous topic when we spoke about DNA, the code of life, we spoke all about the chromosome. And we see that chromosomes are made up of DNA. The genes are sections of DNA. Most of the time, DNA is a long strand called the chromatin network. Just before meiosis and mitosis, the DNA replicates, then coils to form a duplicated chromosome. So you'd have remembered in DNA replication, originally we had a single chromatid, but that chromatid underwent DNA replication, and the two chromatids became joined to each other by a centromere to form an entire chromosome. So this is basically what we will have, and our DNA is going to be found on the chromosome. When we revised mitosis in the previous video, we said that mitosis firstly involves interphase, prophase, metaphase, anaphase, as well as telophase. Now, when we start describing the process of meiosis, we will first look at the phase that's going to require us to have a general understanding of the following term, which we refer to as homologous pairs of chromosomes. Now, firstly, if you take a look at the picture on the right-hand side, you have a green chromosome and you have a pink chromosome. Let's try to identify three characteristics that's going to be common between these two chromosomes. Number one, you notice that they are exactly the same size. The pink and the green chromosome has the same size. Number 2, you notice that the centromere position is exactly the same. The centromere position is exactly the same. The shape of the chromosomes is also the same. Now, purely because these two chromosomes have the same size, same shape, and same centromere position, you can simply refer to them as being homologous chromosomes. But you notice that each chromosome has its own color. And why is that so? When you have homologous chromosomes that are simply going to have the same shape, size, and centromere position, the one chromosome will come from the mother and the other chromosome will come from the father. Some general characteristics to remember: chromosomes occur in homologous pairs. In humans, there are 23 homologous pairs, remember, because there are 46 chromosomes. 23 chromosomes come from the mother, referred to as maternal. 23 comes from the father, referred to as paternal. And together, we have a total of 46 chromosomes. The same characteristic is coded for on each homologous pair, for example, eye color. So what do we mean by this? If we're looking at the characteristic for eye color, on the green chromosome, if we saying eye color sits here on top, then it's going to sit on that exact same position on the pink chromosome. It's going to be the same position right through. So thus far, we provided a definition of meiosis. We spoke about the site of meiosis in plants and animals. We spoke about the fact that meiosis is a continuous process. We are now briefly going to describe the events of interphase. Now, you will notice that especially in your notes as well as your textbook, interphase is not really highlighted as one of the phases of meiosis I. However, you need to remember that interphase takes place before meiosis I or meiosis II. Now, when we are describing the entire process of meiosis, we are going to divide it up into meiosis I as well as meiosis II. I'll repeat that. You have meiosis I and meiosis II. But before meiosis I, you are first going to have interphase taking place. So what actually happens in interphase? Firstly, DNA replication takes place. Why? Chromosomes, which are single threads, become double. So originally you have a chromatid, but they need to become double. So that each chromosome will now consist of 2 chromatids that have been joined by a centromere. DNA replication helps to double the genetic material so that it can be shared by the new cells arising from cell division. So at the end of interphase, you now have your full chromosomes because originally you had chromatids. DNA replication occurred, which allowed the chromatids to double, and they are now joined by a centromere. We can now move on to describing the events of the following phases of meiosis I. We will speak about prophase I, metaphase I, anaphase I, as well as telophase I. So please pay attention carefully because this is extremely important. Your understanding of these phases will form your basis of you being able to answer any exam type question. So if we have to start with meiosis I, we'll firstly speak about prophase I. If you have a look at this diagram over here, on the outside, you can see that you are looking at your cell membrane. So on the outside, you have a cell membrane. On the inside here, you have a dotted circle on the inside. And that dotted line is simply representing the nuclear membrane. And inside the nuclear membrane, you have your chromosomes. And remember, the reason as to why you have your chromosomes is simply because your chromosomes were formed in interphase of the cell cycle, where DNA replication occurred and the 2 chromatids became joined by a centromere. So what exactly happens in prophase I? Firstly, your nuclear membrane or your nuclear envelope is going to start to disappear and disintegrate. Secondly, your spindle fibers are going to start forming. Thirdly, and most importantly, you're going to have a very important process taking place here between these chromosomes. Firstly, you're going to have homologous chromosome pairs lying next to each other. Homologous chromosome pairs are going to come and they're going to lie next to each other. When they lie next to each other, they are going to form bivalents. Your chromatid of your maternal and the chromatid of your paternal chromosome, they are going to eventually meet and they are going to touch and overlap each other. And that point at which they are going to touch and overlap each other is referred to as chiasma. This process is known as crossing over. And crossing over is going to help to increase genetic variation in the gametes, which causes genetic variation in the offspring as well. Let's speak about the important points that occur in prophase 1 again. Firstly, the spindle fibers start to form. The nuclear membrane and nucleolus start to disappear, therefore it's depicted by a dotted line on the outside. An important process of crossing over occurs. Please ensure that you know all the events of prophase because you will be required to describe the entire process of crossing over. Now, please ensure you are able to describe the entire process of crossing over and you know all the points of crossing over. So firstly, you have homologous pairs of chromosomes lying next to each other. And when they lie next to each other, they're going to form bivalents. Remember that one chromosome comes from the mother and the other chromosome comes from the father. Therefore, we have a black and a white chromosome. Secondly, your chromatids from a homologous pair touch. And when they touch, they meet at a point. This point is referred to as chiasma, also known as chiasmata. This is the point where your chromatids are going to exchange genetic information, and that is now going to increase the chance, it's going to increase genetic variation inside the gametes, therefore increasing the chances of survival in the offspring. We can use this diagram to have a look at exactly what occurs in the process of crossing over. So just again, on the left-hand side, you have a red chromosome. On the right-hand side, you have a blue chromosome. The one comes from the maternal, from the mother, and the other one comes from the paternal, from the father. You're going to firstly have homologous chromosome pairs lying next to each other, forming bivalents. Your chromatids, one chromatid from the paternal, other chromatid from the maternal, they're going to meet and they are going to overlap at a point referred to as chiasma, where they touch each other. They're going to exchange genetic information, and when they exchange genetic information, that process was referred to as crossing over, where they touched, and therefore the new resulting chromosomes that you are going to have is going to have different genetic information compared to what it originally had. So now if we look at the red chromosome, it has a full red chromatid, but the other red chromatid has a bit of blue on top and at the bottom because crossing over occurred here on top and crossing over occurred at the bottom. If we look at the blue chromosome, there's a full blue chromatid, but on top we have a bit of red. Why? Because crossing over occurred there. At the bottom we have a bit of red. Why? Because crossing over occurred there. So therefore the new chromosomes has exchanged genetic information, and therefore you're going to increase genetic variation in the gametes, which is going to increase the chances of survival in the offspring. If we now move over to metaphase 1, in metaphase 1 you will see that you still have your homologous chromosomes, but your homologous chromosomes are now going to be arranged on the equator of the cell. And when they arrange themselves on the equator of the cell, they are going to be doing this in a random way. This random arrangement of homologous chromosomes on the equator of the cell in metaphase 1 is going to help to increase genetic variation in the gametes, therefore also increasing the chances of survival in the offspring. So in metaphase 1, your chromosomes line up along the equator in homologous pairs, very important. One chromosome on either side of the equator, and this is a random arrangement. Now, after we've described these two phases, you've noticed that in prophase 1 as well as metaphase 1, there were events that occurred that helped to increase genetic variation. You need to be able to describe each of these events. So number 1, firstly, in prophase 1, the process of crossing over. You must be able to describe that. In metaphase 1, you need to know that the homologous chromosome pairs are going to arrange themselves on the equator of the cell, and it's going to occur in a random way or independently. and this is therefore also going to increase genetic variation in the gametes as well as the offspring. If we now look at anaphase 1, you will see that your homologous chromosome pairs have now separated. So your full chromosomes are going to move towards opposite poles. So you see the black chromosomes are moving to the one pole and the white chromosomes are moving to the other pole. So a recap, in anaphase 1, the spindle fibers are pulling the whole chromosomes towards opposite poles. They've been pulled by the spindle fibers. The homologous pairs therefore separate, and you're going to have the halving of the chromosomal number. This point will be more clearly understood in the next phase, telophase 1. So what occurs in telophase 1? You now clearly notice that that one cell that you originally had starts to divide into two cells. So your unpaired chromosomes have reached the opposite poles, as you can see here. Each pole has only half the chromosomal number. So originally there was one cell with 4 chromosomes, but now each cell after it has split is going to have half the chromosomal number. So instead of 4 chromosomes, you are now going to have 2 chromosomes in each cell. Cytoplasm divides and cytokinesis occurs. So this division of that one cell into 2 separate cells is referred to as cytokinesis, which has formed your chromosomal number to half from 4 chromosomes in one cell to 2 chromosomes in each cell, because that one cell has now divided into 2 cells. That brings us to the end of the description of the process of meiosis I. What you need to remember is at the end of telophase 1, your chromosomal number has now been halved, and it will never ever halve again. So from now all the way until the end of meiosis II, until the end of telophase 2, you are going to keep that chromosomal number of 2 because the chromosomal number has already been halved. So in summary, in this video, we defined meiosis. We spoke about the site of meiosis in plants and animals. We spoke about the fact that it's a continuous process. We described the events of interphase. We also described the events of meiosis I, namely prophase 1, metaphase 1, anaphase 1, as well as telophase 1. In the next video, we will be describing the entire process of meiosis II. So please stay tuned for the next video, where we will be describing the process of meiosis II.